Alrighty, so we're going to talk about Unit 3. It is 3 chapters in the book, and it is 3 big topics: enzymes, photosynthesis, and cellular respiration. So there are 84 slides, and I'll go pretty quick. So we talked about enzymes a few times this year. Enzymes, the thing that you have to remember is they are specific, and they speed up a reaction. So we have enzymes and we have substrates, and they fit like puzzle pieces. And then when they are present, they can make a reaction go faster. And so there are spots on the substrate and enzyme where they come together, and then it makes it so that the reaction works better. We call them transition states, or it puts it into a transition state by modifying the active sites. Sometimes it makes it fit better together. Sometimes molecularly, the pieces that are supposed to be next to other pieces or react with other things are in the right location. And sometimes it bends it a bit to make the structure work a little bit better. And then this is just a figure from your book that talks about the active site is where the reaction takes place, and that's usually where they fit together for the enzyme and the substrate. And then another thing we talked about was cofactors and coenzymes, which we'll get to in the next few slides. But then we'll also talk about endergonic and exergonic as we go through. Just some examples of some cofactors. This is from your book. You don't need to remember this, it's just sort of a gee whiz kind of extra stuff. And then we have cofactors and we have coenzymes. So cofactors are atoms or molecules that are necessary for enzyme function. And then coenzymes, there's a little qualifier in there, organic. And so cofactors may not necessarily contain carbon, but coenzymes will. But they both have basically the same function, is to help the environment be as friendly for the reaction to take place as possible. And so this is just talking about a specific example that has to do with catalase. You can read through that. And then just a picture to go with it. And then ATP is a thing that needs to be present that contains carbon that is necessary for a reaction to occur. And so it's kind of a weird, it seems weird that we would classify it in such what seems like such a small functional little category of things, but that's where it falls into. And so we talked a lot about ATP, but we have an adenosine, 3 phosphates, and then we put the phosphate on or take the phosphate off, and then it's called phosphorylation or dephosphorylation. And then it goes between ATP and ADP. The energy is stored in the bonds and all that stuff that we've talked about. Just some pictures to go along with that. And then sometimes they don't fit perfect, but they sort of squish together for long enough in order for the reaction to occur. And so it's called induced fit. Anytime something is induced, kind of like pregnancy, it's forced to happen when maybe it wouldn't happen easily. And so then it makes it so that there are ways that we make the environment, like I said before, sort of friendly for the reaction to occur when maybe it wouldn't be, or would be less conducive to a reaction. And then there are 3 big things that affect enzymes that we talked about: temperature, pH, and salinity, which is salt content. And so our bodies, we have sort of the normal body temperature. We have, we want to be about neutral pH. We have a certain level of salinity in our bodies. So a few ways to think about that is like we have normal. If you get a fever, that's bad. If you are too cold, like hypothermic, that's bad. And then if you're too acidic or basic, because of oftentimes health conditions can sort of make our blood wacky, and so it affects how the enzymes work. And then one way that you can think about salt is if you've ever heard of an IV and a saline solution, that saline is salt. It comes, and then salinity, it's all the same root word. And so we have to have a certain amount of salt in our bodies for it to function properly. And so if you get an IV, that salt solution is exactly like our normal saline level is in our bodies. So it's pretty cool. And then this just talks about the enzyme activity at certain or for a couple of different kinds of bacteria. So for this one, the blue line, or the E. coli, works at about 38-ish degrees Celsius, which is about our body temperature, which is nice because we have E. coli in our intestinal tract. And then Thermus aquaticus, just from looking at the word, it sounds like something to do with heat. And so it works better at a little higher temperature than us. So if you look at that graph, that's what that's showing you. And then just another example of different things and different enzymes. And this one has to do with pH. Pepsin is stuff that's in our intestinal tract, in our stomach. And so you should know that in your stomach there's acids, so it would work at a lower pH, and then the other two are sort of at regular outside of the stomach parts of the body where they work. And then we talked about this several times as we go through, but shape equals function, and if the shape is wrong, the function is wrong. And so proteins fold. There are 4. We talked about those, I think, in one of the previous units. The 4 ways that they fold. There's primary structure, secondary structure, tertiary, and quaternary structure. But if we change that because of denaturing, then we mess with the function of the enzyme. And so that's what this is talking about. The heat, the pH, the salts. This adds in detergents. It messes it up so that the proteins don't stay bound together like they are. We call that denatured. And so our metabolism, everything in our body, and then these are talking specifically about energy related things, everything is controlled by and paced by enzymes. And so the other part of sort of the enzyme third of these notes is energy. And so a lot of this is pretty easy. So energy is the ability to do work. We have kinetic potential. The 2 laws of thermodynamics. The first one is we can't destroy or make energy. We just sort of transform it from something else into something else. And the entropy is the tendency of a system to go towards disorder. And so second law of thermodynamics talks about is the one about entropy. And so then this is just talking as you add it, the entropy would increase. And then that little thing over on the right hand side is just a little key as to what that is. And then if we increase time and sort of add heat, then it would disperse. And so the entropy would increase, or the disorder of the system would increase. And then this is a little bit about like food pyramids. We only really pass along 10% of the energy of the lower. If you were to think of it like a pyramid, the lower you only get 10% gets passed. So there's a lot of grass and only like 1 eagle, because there's the things in between sort of the dominant predator of a system. There's only a couple of them because the system can only sustain so much because of the loss of energy between what are called trophic levels. And so this is talking about how energy comes from the sun, gets used through, and then it sort of gets lost into the system, and then it's replenished by the sun, over and over and over. And then we store our energy in bonds. Then this is super easy: reactants, products, and a reaction. I don't even need to explain that. But this just gives you an example of a little chemical reaction. And then if we store energy in a bond, we call it endergonic. If we break a bond, it's called exergonic. I think it sounds exactly what the words mean. Exer means out, ender means in. And then this is just showing an example with the same little molecules just to show. And then we have different pathways in our bodies. Some of them break things down, some of them put things together. So anabolic puts them together, it builds things. I remember this because like anabolic reminds me sort of like steroids, and steroids build muscle. And then catabolic is breaking things apart. And cats, like my little joke in class was cats knock stuff off of shelves and break stuff. So cats break and anabolic builds. And then when it's a cyclic pathway, and we'll talk about this more when we get to cellular respiration, it's a cycle. And so the stuff that comes in, the stuff that goes out, we sort of regenerate what we started with. So then it's called a cycle. And then there are certain ways to prevent something from happening or start something from happening. And so sometimes there are, say we need a molecule to do something, an enzyme to do a reaction, and then we make a bunch. And so that bonds onto a spot that would prohibit more from being made. So we only really make as much as we need, and then say we use up all the sort of stuff that's flowing around, and then we use that one that got bonded on there to inhibit, and then the system, that little sort of like or detach of the one, allows that to then go and start the enzyme production again. So it's just a way that we sort of control, and it really comes down to energy use and wasting resources. Our body does stuff as efficiently as it can. And so then this is just a picture of that. And then just some pictures. And then I like this picture because it puts sort of everything in one thing. Most of the time in our body, not all the time, most of the time, we do things that are called coupled reactions. We generate some energy so that we could use that energy to do a job from. And so this is sort of, we release the energy and then we gain some energy. And then we have some reactions that store energy, but then later on they're broken down to be used. And then so it's sort of over and over and over. And so this just sort of puts all that vocabulary in a nice little graphic. When we have, let me skip forward. Okay, good. When we have a reaction in our bodies, we have what's called activation energy. You learn about this in chemistry. You have to give a little extra to sort of give the oomph in order to get the reaction going. And so this is a way that our body controls the release of things, the reaction of things. And so it just makes our bodies work conservatively, I guess would be an okay word. And then this just talks about a specific coupled reaction. So photosynthesis and cellular respirations, they are two things that sort of feed into and off of each other and give something and make something and vice versa. And then this is just talking about the specific little part of that. And then same thing on this one. And then oxidation reduction reactions are a thing you might have talked about in chemistry, but I remember these because something is reduced because it becomes more negative. And so when it is more negative, it has gained the negative particle. And so the portion of the molecule that gets reduced, or the portion of the reaction that gets reduced, is the one that gains the electron. And so the one that loses the electron, it's losing negativity. And so it becomes a little bit more positive. We call that oxidized. And so then this is just a picture of some reactions that do that. And then there's this, which we'll get to. And then we have photosynthesis. So chapter 6. And so we have autotrophs make their own food. Photosynthesis is a process where we use sun. Heterotrophs eat things that make their own. This is just a picture of the visible light spectrum and then the whole electromagnetic spectrum just for a reference. And then we care about pigments because they absorb the sun. We specifically care about chlorophyll, the pigment inside the chloroplasts, because that is the in plants, that is what has evolved to capture, and the location of photosynthesis to capture that energy. And so we have different pigments, there's different colors of leaves and different things like that. But the one that is the most useful is chlorophyll. And so we have two stages. We have light dependent and light independent. So light dependent means it needs the sun. Light independent means that it does not need the sun. And so sometimes if you're studying different things, it might talk about the light and the dark reactions. I don't like those. But also sometimes you might hear about photosystem 1 and photosystem 2. Photosystem 1 is actually the second thing that happens, but it's the first thing that was discovered. And then photosystem 2 is the first thing that happens, or light dependent, but it was the second thing. And so that's just why they named it that. Kind of weird. This is just the equation. So the easy way to remember this is what does a plant need in order to grow? It needs carbon dioxide, it needs water, and it needs the sun. And then it makes glucose, which is the structure of the plants. And so then we eat plants and get glucose. And so we're eating what it made, and then it gives off oxygen. And it gives off water, but we don't really care about that. And then some structures that we care about. The chloroplasts are the organelle that holds the chlorophyll. So chloroplast, I think of it like a plastic container, like a Tupperware or a Gladware, and so it holds it. The thylakoid membrane is the membrane inside that we can build up a gradient past. We'll get to cellular respiration in a sec, but whenever we talk about membranes and energy, it's usually talking about a concentration gradient of something. And then the stroma is just another area of the plant. And so this is a picture just showing you those things. So the stroma is that open space. The thylakoids are like little green coins, and they are green because they are filled with chlorophyll, and then that is all contained within the structure that we called a chloroplast. And so this I talked a bit about photosystems. And then this is just talking about how we put the ADP and inorganic phosphate together because the sun, the energy that came in, we stored it in there. It also breaks apart the water. And then we take that oxygen and we release it. And then we take the hydrogen and then we scooch it over to the second part of the system, into photosystem 1, or the light independent reactions. And then the carbon dioxide that we bring in into that cycle, then we add it all together and we make glucose. And then this is just talking about the non-cyclic. So this is photosystem 2. And so it is the part where the sun is in the picture that I drew. It was the stuff on the left-hand side. It's the first thing that happens. And then this is just talking about how we take the hydrogens and we have a couple of little couriers, or one little courier, NADPH, and it carries it across and it takes it into the cyclic pathway, and then it adds it up with carbon dioxide and we make glucose. So this is just talking about those things. If you remember, I talked about how the little like cart guys at the Pepsi Center, and they go and they pick somebody up and they take them over and they drop them off, and then they come back and then they pick up another one and they take it over and they drop them off. That's sort of what NADP plus is, and like at the Pepsi Center, you might be the hydrogen, or in our bodies or our plants, the H is an H. And then photophosphorylation is light is phosphorylating, or light is adding a phosphate onto the ADP. And so the energy that's coming from the sun, we store it into the ADP and inorganic phosphate, and then we get ATP. And then this is talking about the cycle on the photosystem 1, or the light independent reactions. It's called the Calvin-Benson cycle. And so it sort of does some spins. We bring the hydrogen over from when it is cleaved from the oxygen from water, bring it over and then add it in with the carbon dioxide that the plant breathes in. And then fixation means you are attaching it. So like a glue is a fixative, or you affix something. And so carbon fixation is when we take carbon and we attach it to something. And so we're taking carbon and oxygen and the hydrogen and adding it all together, making glucose. And then we don't talk too much about the nitty-gritty of this stuff, but Rubisco is a pretty significant enzyme that is necessary in order for this to take place. So if we take a second and we look at carbon dioxide, and then we think about glucose, which is 6 carbons, we have to somehow get that stuff connected. And so when we talk about carbon dioxide, or when we talk about cellular respiration, we'll talk about taking a 6 carbon sugar, breaking it down into a 3 carbon sugar, breaking it down into 2 carbon sugars. This is sort of going in the opposite direction. So the enzyme that does the work to put the things together is called Rubisco. And it's in the light independent reactions, which is a cycle. That's why on all the drawings you'll see it's a little circle. And then here's just a picture of it and some many details. And then the stomata is a little, it's like a plant mouth. And so it's where the gases get exchanged, and it's where the water can evaporate from. And so it's pretty cool. We can close them or we can open them, usually depending on how hot or dry or something like this to sort of conserve water. But pretty cool. Here's a picture. But there will be in a second. And then photorespiration is that process. And then this is a picture that I gave you in class and we labeled it. So the left-hand side of this picture is the light dependent reactions, or the photosystem 2. And then the right-hand side is Calvin cycle or Calvin-Benson cycle, or photosystem 1, or light dependent reactions. But you can see how the light comes in, the water comes in, it breaks it down, the oxygen goes out, the hydrogen goes across into the right-hand side, and then take the carbon dioxide from the top, incorporate that hydrogen, and then end up with glucose. So now we'll talk about cellular respiration, which is kind of the opposite of photosynthesis, but kind of not. And so this is a mitochondria. It's pretty exciting, but look at all the squiggly membranes because that's significant. And so we have 2 different pathways when it comes to cellular respiration, and really metabolic pathways, period. Aerobic and anaerobic. Aerobic, you need oxygen. Anaerobic, you do not. And so we have evolved to do aerobic respiration because it works, because it is efficient, because it gives us a lot of energy. And so if we think about, and we'll get to this in a bit, but if we think about the different parts of cellular respiration, the first little chunk is glycolysis. Glycolysis is anaerobic, and it only gives us a net of 2 ATP. That's not very much. And so we have, if you remember from a unit or 2 ago, we talked about the endosymbiotic theory, or endosymbiosis, and I talked about how mitochondria were a little sort of self-sustaining kind of organism, kind of like a bacteria, and we sort of hijacked them, got them into our cells, and then they make energy. Now they depend on us and we depend on them. But when we sort of incorporated that into the very early precursors of now what we think of as bacteria, it gave those cells the ability to produce more energy, and with that more energy gave us the ability to become differentiated and specialized and fancy and smart and have brains and hair and skin and a whole bunch of other stuff. And so this just talks about the 3 stages of aerobic respiration, or what we call cellular respiration. And so we'll talk about each one of them. But there's the formula for cellular respiration. And so this talks about, it pictures it like coins because it's like currency, but I only care about the money, or about the number of ATP. And so it takes a little money to, got to spend a little money to make some money. And so we end up with 2 ATP in glycolysis, 2 ATP in the citric acid cycle, and then 32 ATP in the electron transport chain. If you look at different books or sources, sometimes it gives you different amounts, but we'll just stick with these because that's from the book. And then aerobic respiration is in the presence of oxygen. Oxygen is present inside the mitochondria, which is why mitochondria are significant. If it happens outside of the mitochondria, which is glycolysis, then it is anaerobic. And then we'll talk about fermentation a bit at the end. And so the first thing that happens in glycolysis is glycolysis. It means that you are taking the 6-carbon sugar and you're breaking it down into 2 3-carbon sugars. And then this just talks about you have to spend a little to make a little, so you have to invest a little money, a little bit of energy to get that broken down because it takes work to break that bond. And then this just talks about the details of that, but we call that substrate level phosphorylation. We are phosphorylating, or we are making the ATP at the substrate level, at the chemical reaction level. So enzymes and substrates. And so when we break that bond, that is a small localized little reaction. And so that's why it's referred to as that. And then once we get the 2 3-carbon sugars, which are called pyruvic acid or pyruvate, they go into the mitochondria, and then we break those down in the second one. Sometimes you'll see it as the Krebs cycle. I usually call it the citric acid cycle. It's a cycle, which means there's going to be a circle, which means we are going to regenerate something that we are going to use. And then we're going to take those 3-carbon sugars and break them down into 2-carbon sugars. And then that carbon, that sort of lost carbon, so we have the 1 here and the 2 here, these go in the circle. So this is a 3-carbon sugar. We broke it down into 2 2-carbon sugars. This carbon gets released as carbon dioxide. And so, excuse me, if we have 2 pyruvates coming in, we're going to spin around a few times, and then we're going to get some things released, and then we're going to get some hydrogens freed. Then we're going to pick those up with our couriers, just like we did in photosynthesis. And so then we have this NADH is a thing on here, and we'll also talk about FADH. It's the same thing, like the little courier at the Pepsi Center comes and picks it up, and then this one takes it and transfers it over to a different part of the mitochondria across a membrane, and we're going to build a gradient, and we'll talk about that in a bit. And so the Krebs cycle gives us, for each little zing, so we have a pyruvate, and so on here, for each one, it gives us a certain amount of things. So it gives us 3 NADH and 1 FADH2. Now thinking about that, that gives us the possibility of 4 H's, if you take a second. And so it's going to happen twice. That would be 8 H's for this little chunk of things. And then we get an ATP for each thing, and then we get a net ATP of 2. Actually, so it would be 4, but anyway, hopefully that makes sense. It's a lot of math, but really we care about the hydrogens that are getting broken off and then carried over to a different part of the mitochondria. And so then we take we broke the glucose, we broke the pyruvates, and so what happens is these hydrogens get released, grabbed by other things, and then transferred down. And then what happens is they get dropped off at little spots on the membrane, and then they get pushed across the membrane. And so then we have a whole bunch of hydrogens piled up on one side of a membrane. And so we're building up a gradient. And so this talks about the different pieces and parts that do that. And so we have a whole bunch of hydrogens build up on one side of the membrane, just wanting to come back in. So they're like out there and they're ready. And so they come through in a very controlled way through a specific enzyme. It's called ATP synthase, the enzyme that synthesizes ATP. And so then we have ADPs and inorganic phosphates, and then the energy that is carried with the hydrogen ions that comes back across, attaches those together, and then we get ATP, and we get a whole bunch. So this is just a picture. We had this in class and we labeled this, and we talked about things that get broken down, things that get made. And so on this, I talked about it for glycolysis, is substrate level phosphorylation for that. It's called the same thing in the citric acid cycle because it's just breaking things down. We broke down the pyruvates, and so that breaking is local. It's very controlled. It's right there, so we call it substrate level phosphorylation. And then oxidative phosphorylation, which is behind the little square where I am, but it's also written in the purple square up above it, is when we use the gradient. And it was also called chemiosmotic phosphorylation. So when you think of all those words and sort of break them down, it goes from an area of high concentration to low concentration, and then we make something from it. Phosphorylation is, what are we doing? We are phosphorylating, so we are making ATP. And so then sort of a backup, but also some organisms only use this, is fermentation. And so we can ferment and use a little bit, but it's not efficient, so it's not how our bodies normally function, but it's sort of a backup plan. It's also the only thing that things like some yeast and bacteria, what they do in their normal daily life. So this is just talking about how we make certain things that we have to break them down in order to get the sort of big bang out of them that we want. And if there's no oxygen, we can't do that. But we can still get that too little, like 2 ATPs. It's not really great, but we can use it. And then there's two kinds of fermentation, and the way that they're differentiated is the organism that does it. Alcoholic fermentation is yeast and bacteria, and then lactic acid fermentation is like animals and eukaryotes and different stuff like that. And so that's just, it's what the product is. And so our product is lactic acid, and the for prokaryotes and certain yeasts and stuff like that, the product of the reaction is alcohol. And so this is just talking about how this all took place when the atmosphere contained oxygen that was O2, like we breathe in. It wasn't always like that. Sort of the formation of the universe and the formation of the planet and all that kind of stuff, it wasn't always available. But when that happened is when organisms started to evolve into bigger, more complex things, because we were able to utilize the atmospheric oxygen, and then evolution, we got something that gave us a whole bunch of energy. And so then this just talks a little bit about the carbon cycle, which is just a little aside, so not very much, but it talks a little bit about CO2 in the atmosphere and things, and break it down. So just read through that. But that's it.